Hi, this is Roger Green, host of the Surfing the MASH Tsunami podcast. This week, we're offering 5 conversations from episode 20, our review of drug development stories from the EASL Congress 2024 with Jörn Schattenberg, Naim Alkhouri, and Mazen Noureddin, plus from the vault, one of our wrap-up conversations from the EASL Congress 2023. This conversation focused on 2 specific classes of drugs discussed at the EASL Congress, the GLP-glucagon combinations and the FGF21s. Previously, key opinion leaders on this panel all stated they had greater confidence in the GLP-glucagon than in GLP-GIP. Now, Naim goes deeper into this issue by noting a key issue and difference between the agents. The GLP-1 to glucagon agents have different ratios between the 2 substances. Since glucagon increases blood sugar, this ratio can reflect value in diabetes as compared to ability to drive liver behavior. He goes on to mention specifically that the efinopegdutide study included a semaglutide cell, which showed further reduction in liver fat versus the GLP-1 agents alone. Shifting to safety, Jörn comments on the high level of tolerability in the survodutide trials, and Mazen shares his high hopes for GLP-glucagons and also for triple agents earlier in development. He ends this section by sharing some of the survodutide data from the New England Journal article. From there, the conversation shifts to FGF21s. Mazen provides data from the late breaker that Vlad Ratziu presented on behalf of Stephen for efruxiermin. This is a 96-week triple biopsy study showing dramatic sustained fibrosis improvement with a 30% delta for 1 level reduction and a 21% delta for 2 level reduction. Jörn notes that the triple biopsy design demonstrates the durability of this agent against fibrosis. I comment this is particularly encouraging because of the previous pegbelfermin data showing loss of efficacy and tachyphylaxis. Naim concurs and adds that if the 2-stage drop in fibrosis can be sustained, this would be, and I quote again, a big deal for the field, unquote, and might lead prescribers to think about FGF21 as an induction therapy using a more tolerated oral agent later on as maintenance. During the episode, all of us kept comparing the negativity and virtual despondence of the 2019 to 2020 period described as the graveyard with what's described as today's gift shop of multiple modes of action, demonstrating clearly positive results. It's an exciting time for the field and for the patients. As I edited, I found myself thinking of Stephen's goal to put a big fat dent in fatty liver disease. We all can use this episode to gauge the size of Stephen's dent, and frankly, it's pretty damn big.
Naim Alkhouri.
To speak to the servo data, a very exciting mechanism of action, dual agonist. And again, the glucagon has shown direct hepatic effects. And you talked about the Merck product, efinopegdutide. What I liked about their study is that they compared it to semaglutide and they showed further reduction in liver fat on MRI-PDFF. So again, showing you that adding glucagon has the direct liver effects. I presented data at the DDW looking at the NIT data with survodutide after 28 weeks of treatment, showing nice signal on ELF and ProC3, 2 fibrosis biomarkers. And then the EASL presentation confirmed on histology that there is improvement in fibrosis. So it's a big deal. I think one thing to keep in mind is when you add glucagon, glucagon is something we use to treat hypoglycemia. So it can actually increase your blood sugar, which is not something we want in this patient population. So that ratio of glucagon to GLP-1 is potentially a very important thing to keep in mind. And the distribution of this ratio, if you start with survodutide, it's actually 1:8. So 8 GLP-1, 1 glucagon. So kind of like just a touch of glucagon with this compound. And that's why there's actually reduction in A1C. So the way I think about it, it's really a potent GLP-1 with some glucagon to have liver-directed effects. If you look at efinopegdutide, the ratio is 1:3. So 3 GLP-1, 1 glucagon. And then pemvidutide is really 1:1. So that's why you lose improvement in A1C with pemvidutide, but the question is it going to have more liver-directed effects compared to survodutide? Do you need more targeting on the glucagon? So these are questions that we will answer now. We have the phase 2b pemvidutide trial. I will have histology data to compare, and we'll see if they achieve higher rates of MASH resolution and fibrosis regression. But I think with the data we have some direct comparison to just mono GLP-1. It seems like glucagon is doing some additional work in the liver that you don't see with just a GLP-1.
Jörn Schattenberg.
For all of these trials, and I mentioned that in my previous comment, I think it's important to keep the patient on drug, right? To give them a dose that's tolerable, and then over time develops an effect with preferentially a liver-targeted mechanism of action, I think having the strongest rationale, and particularly in advanced fibrosis. The one piece of data we didn't discuss yet was there was a safety study in compensated and decompensated cirrhosis on survodutide, and even in that population there was improvement of NITs and tolerability was robust. So that was an oral presentation at the MASLD therapy session given by Dr. Lavis. I think the companies are learning about how to stepwise increase and figure out the highest tolerated dose. And as a concept of maintaining patient on drug, that dose, I think, is crucial. And that's going to be how we do it in clinics. Then, if you have the drug, patient doesn't tolerate it, you can stop, maybe pause at a lower dose or titrate down, and then try to continue and keep them on drug.
Mazen Noureddin.
I mean, I really have big hopes for the dual GLP-1 and glucagons. To mention, there's a triple. We don't know what's going to happen with it. They're playing with it in the obesity and the diabetes world, more in the obesity. So one thing goes for Lilly is they don't just have tirzepatide, they have retatrutide. And I don't know what's their plans in terms of development of retatrutide. It will be very interesting to see what it does in NASH if they try to explore what is it in NASH. I look also, we need to keep in mind there might be side effects with these drugs beyond nausea and vomiting, and we have to keep an eye on the heart rate and all these things. Going back to survodutide, I think with the GLP-1s and the duals, it is interesting to look at data in different ways, completers, paired biopsies, as well as intention to treat. I've heard some people that they got confused because that's not traditional way, but that's what also they do in the obesity trials. But to talk about survodutide in particular, Jörn, Naim, and I are all co-authors on that paper that came out in the New England Journal. And the abstracts actually spell out I believe that's the ITT numbers. And the fibrosis improvement, 36 compared to 22%. That's the highest delta, and actually 62% on NASH resolution in the most effective dose compared to 14%. If you look at it, paired analysis, if you look at it completer, the delta even widened more. And if I recall correctly, no matter how you look at things with survodutide, there was dose dependency. Of course, when you get to the 6 milligram, you got a little bit more dropout because of the side effects. But there was a clear message that they get NASH resolution, fibrosis improvement with statistical significance, regardless of the way you look at the analysis. Clearly, like a very efficacious drug. There are plans for phase 3s and different patient populations. So I'm excited that PIMV is, hopefully they will show us the data sometime next year. Just go back a little bit to survodutide. There's also additional NITs data that are going to be presented at the AASLD that we're looking at right now, in addition to data that Naim presented. I'm really excited about efino and pemvidutide. I think they're going to come from pemvidutide next year. So as Naim mentioned, excited 2024, and it's going to even get better in 2025.
Yeah, and we haven't even gotten to the FGF21s yet, have we?
Yeah, I mean, let's go to it. I'm happy to take a first jab. I think this was presented at EASL, and we have been working on the publication. The paper was presented by Vlad on behalf of Stephen. And again, efruxiermin is an FGF21 that is currently on a phase 3 development, including all patients population, pre-cirrhotic and cirrhotics. Because efruxiermin is another FGF21, also on phase 3 development. But here, efruxiermin continued to excel compared to many other drugs, as well other FGF21s, when they showed us 96-week data showing brilliantness in design and just looking at the next steps, not like really short terms. And here they have biopsy data up to 96. And the elephant in the room, that the amount of fibrosis improvement by one stage, which was presented as up to 75% compared to 24% in the placebo, 75 and the 50 milligram, 24% in the placebo. And that was liver biopsy analysis set. If people ask about ITT, and the ITT in that study was on the 50 milligram, 49% fibrosis improvement compared to only 19% on the placebo. That is a 30% fibrosis delta. That's a big deal. Importantly, if you also read between the lines, they looked at fibrosis sustainability beyond 24 weeks up to 96 weeks and by one stage. So if you were a responder and you continued beyond to 96 weeks, the sustained responders was up to 92%. But you also had newcomers at the 96 weeks on those who did not respond previously to 24 weeks, which makes sense, right? Fibrosis takes time, and it was on the 50 milligram was up to 63%. There are many other data that also you can read read between the line. I mean, for instance, the 2-stage fibrosis improvement was up to 23% in the 50 milligram compared to only 2%. And this is important. This is low placebo response. And the reason why it's low because it's 2-stage fibrosis improvement, which is much harder for the placebo to get. And it makes sense to have such a low placebo response in 2-stage fibrosis improvement, not other parameters. Again, 21% to delta and 2 stages fibrosis improvement. NAS resolution, same story. The highest was 62% compared to 24% in the liver biopsy analysis set. If you look at ITTs, was 40% to 19%. Again, another big delta, but there's a clear anti-fibrotic effect. There's clear sustainability in the improvement, and continuation of treatment led to significant. The side effects is not as bad as GLP-1s. And finally, I could be wrong now, looking between the lines, seems to me like efruxiermin, they do this extra steps beyond 24 or longer duration up to 96 weeks. A little bit edge on because of efruxiermin, but I need to see more analysis, and I don't want to compare apple to oranges, but efruxiermin clearly standing high and tall in the field.
The important data point that I felt here was triple biopsies with another biopsy after 96 weeks with an earlier response. So you can actually see this supports the concept of pathophysiology in this disease so strongly. You can see how the response rates go up over time, how most of the patients, I think it was most of the patients maintained. I think maybe one patient that did have response at the earlier time point didn't have it at the later time point. Most patients maintain it, and that's really reassuring. The only other study that I remember with multiple biopsies really the semaglutide, where we saw it going up and down. And I think here with a stronger mechanism, we're really getting reassurance this can be dealt with over time.
Roger Green.
Well, yeah, and then here, if I recall correctly, particular burden, because the pegbelfermin work suggested that there might be efficacy that might recede by 48 weeks. So that being the case, we now know that would have been, if the case was idiosyncratic to the molecule or to the medicine and not to the class, which is, given how good the class is, that's a really exciting thing too. Naim, you look like you had something you wanted to say, or did I get that wrong?
I was going to say the exact same thing that you just said, Roger, that, this was reassuring for me because of the pegbelfermin and that tachyphylaxis concern that we had with FGF21s. And, Mazen summarized this very nicely, but, the patients that responded at 24 weeks maintained the response. So that was very reassuring because what we saw with the semaglutide, it was the seesaw effect. So the responders on the second biopsy lost response on the third biopsy. And then we had more, newer responders on the third biopsy that were not responders on the second biopsy. So all these, hypothetical concerns in my mind were put to rest. Obviously, the numbers are small and we are doing the phase 3 trial now. So this is going to answer all these questions more definitively. But this was my biggest takeaway is that, no issues about losing response after 96 weeks of treatment. And then, of course, that 2-stage fibrosis regression, if that holds in a larger sample size, this would be a big deal for the field because, it takes 10, 15, 20 years to go from F1 to F2. And now with 2 years of treatment, you can bring them back from F3 to F1. That's really remarkable. So I think we'll see if that holds, but that can change the way we think about how we use these medicines. And maybe they will be used as part of some induction therapy to take people from the edge, I guess, and bring them back to a very early disease stage and then maybe transition them to a more tolerated oral medication.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of the conversation or the entire episode, please put them in the reviews section of the page from which you downloaded this conversation or send an email to questions@surfingmesh.com. Next week, Frank Taka and Luca Valenti from the EASD, EASL, EASO combination group will be joining us to discuss the updated clinical practice guidelines for MASL. Until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.